Good afternoon to students. Today we are discussing cell cycle and cell division. So first we discuss cell cycle. In cell cycle there are two main stages. First stage is interphase and second is M phase. So we see this diagrammatic representation of cell cycle. In this stage interphase, so from G1 to G2 it is known as a interphase. Interphase means it is a preparative phase. It is a preparation for M phase, and in interphase there are three substages. G1 phase, G1 means gap 1 phase, then second synthetic phase, and third one G2 means gap 2 phase. After that M phase. M phase means it is also known as multiplication phase. So here karyokinesis and cytokinesis are the two main stages. Karyokinesis means it is a division of nucleus, and cytokinesis means it is a division of cytoplasm. In karyokinesis there are four stages: prophase, metaphase, anaphase and telophase. These are the actual multiplication stages of cell cycle. Whether it is a mitosis or meiosis, in that these four stages are mentioned. And by these four stages the cells are divided. This interphase, it is a preparative phase for cell division, and after that it goes to the phase M phase. So we see this diagrammatic representation of cell cycle. From here to here, it is a preparatory phase, means it is a interphase. In that first we discuss gap 1 phase, so it is here. Then after that synthetic phase, then gap 2 phase. So let us discuss these stages in detail. G1 phase, here the protein synthesis is take place. So all the enzymes which are required for DNA replication, these are synthesized in G1 phase. Then RNA synthesis also increased in this G1 phase. Then third point is ATP synthesis. All the ATPs are required for the cell division, or the chromosomes which are present in the cell. These ATPs are required. So here ATP synthesis is also increased. Then the raw materials are also prepared in G1 phase, which are required for S phase. Then fifth point is the size of cell. So here if this protein synthesis take place, the protein synthesis is increased, RNA synthesis increase, then ATP synthesis increase. Also the raw materials are prepared in this cell. So as well as the cell growth is also takes place in gap 1 phase. So after that we discuss S phase. S phase means in S phase it is a synthetic phase, and in that original DNA replication takes place. So before that we discuss RNA synthesis or protein synthesis. In protein synthesis the enzymes are prepared that are required for DNA replication, and these enzymes goes through S phase. So here DNA replication takes place. Then centriole is also divided in this stage. Centriole, which is present in animal cell, but it is absent in plant cell. So only in animal cell the centriole is divided. Then histone proteins, which are also synthesized, which are present in higher cryptograms. In higher cryptograms histone protein is required, that is also synthesized in S phase. Then third step, gap 2 or G2 phase. Here also protein synthesis increase. So in G1 also protein synthesis increase, but the tubulin which are required for the spindle fiber formation. The spindle fibers are formed in M phase the stage of metaphase. Here the spindle fibers are formed. So for that tubulin protein is required, and this tubulin protein synthesis is increased in G2 phase. Then plasma membrane proteins. The proteins which are required for the formation of plasma membrane, these are also synthesized in G2 phase. Then ATP synthesis. Suppose when one ATP move from the equatorial plane to the pole. So suppose this one is a cell, and in that cell these two centrioles are present. So it is a equatorial plane and these are poles. If one chromosome is moved from equatorial plane to the pole, for one chromosome 30 ATPs are required. So in human cell 46 chromosomes are present. So one ATP means the one chromosome require 30 ATPs, means 46 into 30 for a single cell after division, and 40 for next cell after division. We know here in mitosis the first cell is mitosis or meiosis, the cell is 2n number, and in mitosis the cell 2n number of cells, two cells of 2n number are synthesized or divide from one cell. Here 46 chromosomes in one cell, so these are parental chromosomes and these are divided, means 46 and 46. These are 2n number, these are daughter cells. So this 46 and this 46 into 30 and 30. So this much ATPs are required for synthesis. So that much ATPs are synthesized in G2 phase. Then next phase is M phase. Here the actual division takes place. The actual division from these 5 stages. In these 5 stages, first karyokinesis. In karyokinesis, first rho phase, metaphase, anaphase and telophase. Here the chromosomes are spindle-like structure, separated structure. Then in metaphase spindle fibers are formed. Then in anaphase the cytoplasm or the chromosomes are separated from the equatorial plane in two halves. From both poles these are divided. Chromosomes are divided towards the both poles, and here sister chromatids are formed. Then in telophase the nucleus is divided in these two cells. Then cytokinesis. In cytokinesis the cytoplasm is get divided. So after that one G0 phase is also present in cell cycle. Let us discuss G0 phase. It is actually a inactive stage. After the division of M phase, the G0 phase is present. So it is also known as a inactive phase. So in G0 phase there are 3 stages. Sometimes these 3 stages are present. First is a differentiation. Differentiation means what? The cell now come out and perform special function they do not get back again in the cycle. If here the G0 stage is present, it is inactive, and this G0 stage or the parenchymatous, here the parenchymatous cells are present, means meristematic cells. These are capability to divide and redivide. So here in G0 phase the parenchymatous cells or a cell is come out this here it is come out from the cell cycle. The cell is come out from the cell cycle and perform a special function, and they do not get back again in the cycle. It is known as differentiation, means it is differentiated from the cell cycle. That means differentiation. Some are the exceptional cases, that means a parenchyma. Here this one is a structure of a plant TS, stem TS. So here epidermis, cuticle is present, and after that these parenchymatous cells are present. The parenchymatous cells become meristematic because they are having the capability of division, and these are after division these are form meristematic cells or cork cambium toward outside. Here the xylem cells are present and here phloem cells are present. The dividing cells are xylem. Then here these are known as parenchymatous cells. The parenchymatous cells are again, these are divide and again come back in the cell. If these are come back in cell again, so these are called as de-differentiation. Differentiation means they come out, and de-differentiation means they come after dividing, they come back again in the cell cycle. Here this arrow indicates the comeback of parenchymatous cells. Then redifferentiation. After division, parenchyma will again go outside of the cycle. It is known as differentiation. Here the cell is divide, means parenchymatous cells are divide again and come back again in the cell cycle. Then again if the parenchyma is again go outside from the cell cycle, it is known as redifferentiation. So students, this is the end of our topic, cell cycle.